Good afternoon. Welcome to Medicine Grand Rounds. As everybody knows, today is Research Day, and we have a special speaker. Our Research Day invited speaker is Dr. Toren Finkel, who is coming from University of Pittsburgh. Dr. Finkel obtained his MD PhD at Harvard and did uh, his residency at the same institution before moving to Johns Hopkins to have his fellowship in cardiology. Then he moved to the NIH in 1992, and he stayed at the NIH for at least 25 years, having different positions at the NIH, including chief of the cardiology branch and chief of the Center for Molecular Medicine at NHLBI. He's a member of the American Society for Clinical Investigation, the Association of American Physicians, and he has been also induced as a fellow of the American Association for Advancement of Science, and is an elected member of the National Academy of Medicine. He has held multiple roles in the research, and then he moved in 2017 to Pittsburgh, where he is the director of the Aging Institute and the Nicholas Beckwith and Dorothy Beckwith Endowed Chair in Translational Medicine at University of Chicago. Sorry, Pittsburgh. And then in terms of his research, his major contributions have been on understanding the role of reactive oxygen species, mitochondrial function, intracellular signaling in the aging context, and how that alters aging diseases. And I really want to thank Dr. Finkel for accepting this invitation, and I really want to ask everybody to join me in welcoming Dr. Finkel. Thank you.
Thank you very much, Ernesto. It's a pleasure to be here, and thank you all for coming. I don't eat lunch, so it's really hard for me to watch people eat at the same time. But I do that for hopefully to live longer, but I'm not sure it will work. But so I'm going to talk a little bit about the biology of aging and maybe give you a little sense of. Okay. Why don't you go ahead? Screw it up. Okay, good. Thank you. While they're doing that, so give you a little sense of the field in general. Then in the middle part, where you can really check your phone and everything, I'll talk a little bit about my own science, and then come back a little bit and talk a little bit about the clinical things that are ongoing at the institute I direct, and what I think potentially could be ways that we could potentially intervene in the aging process in a meaningful way. So I'd like to start off just asking why we should even study this phenomena. And so I think from a scientific point of view, I think it represents the greatest unknown biological question that exists. So we have a pretty reasonable idea about many disease processes. For instance, I was trained as a cardiologist. I think we have a reasonable idea how atherosclerosis starts and develops, how cancer develops. We really have no clear understanding of why and how we age. And if we're lucky, we all do it. And as a biological phenomena, it's really relatively unknown. And there's no model that everybody accepts in terms of why and how we age. So I think as a question, it's a fantastic question. But I think the reason a lot of people are interested in this as a topic is because it's of the intimate relationship between age and disease. And so I'll give you an example, again, because of my background in cardiology, just of the magnitude of which age is a risk factor for disease. So as you know, there's a number of defined epidemiological risk factors for the development of heart disease, but their relative importance, I just wanted to walk through with you. So if your cholesterol is really elevated, above 280, you're based on risk of a cardiovascular event over the 10 years doubles, basically. Similarly, if it triples if your blood pressure is higher, doubles again if you have diabetes, doubles again if you smoke. But if you're over the age of 70, your risk for a cardiovascular disease goes up by 6 or 7-fold, right? So that's the magnitude of risk that age confers versus what I would consider relatively minor risk factors like smoking and diabetes. The difference between these set of risk factors is that you can go into your, you probably get asked this, but people can go in and get, relief and modification of risk factors like cholesterol and blood pressure and diabetes, et cetera. And so we lump those smaller risk factors into modifiable risk factors. And by reducing those risk factors in the population, dramatically have reduced the incidence of cardiovascular disease in the US. But they're relatively small. And the issue is, can we view age itself as a modifiable risk factor? Can we, in fact, think of developing therapies or interventions that slow the rate of biological aging? And if we could do that, given its relative risk, magnitude of risk, what a dramatic impact would potentially have on cardiovascular disease. And the issue is, it's not really just cardiovascular disease. So it's really every disease that we worry about. Here's the incidence of cardiovascular disease as a function of age, but the same thing could be said for neurodegenerative diseases and for cancer. The risk of getting cancer over the age of 65 is 10-fold higher than under 65, and it really dwarfs any other risk factor for that condition. And so, while therapies and institutes at the NIH, or at least they have been sort of thinking about each of these conditions based upon what I would consider the y-axis, trying to get diseases specific for Alzheimer's diseases or specific for cardiovascular disease, attacking, again, that y-axis. They all share this common x-axis, right? And so if you can think about therapies that slow biological aging, you could potentially, and if they are common mediators of aging across all, then you could potentially be thinking about therapies that could impact and slow or prevent a whole range of conditions. So the question is, how far can we push these curves rightward? How far can we slow aging itself? And probably the answer to that is shown here. This is Madame Jean Calment, who, if you disregard things like Moses and Methuselah and other biblical references, she has the longest known recorded lifespan that we know of. This is her on her 121st birthday. She lived almost 2 more years until she passed away just before her 123rd birthday. And I like to tell this story. Some of you may have heard, but so she lived in like southern France, a beautiful place, Arles, and actually her uncle had like a art supply store, so she got to meet all the French Impressionist painters over her course of her lifespan. Had some very unkind words to say about Van Gogh, who she thought was very rude, and et cetera. But in France, if you have a condominium, you can, in a way, sell that to someone in advance of your passing, and they pay you a sort of a small monthly fee with the idea that when you pass away, that they get your apartment. And so when she was in her early 90s, this very nice gentleman who coveted her apartment in his 40s began paying her this monthly retainer to inherit her place. And as you can imagine, he died in his early 70s before he could move in. But anyway, so the question is, so did she do anything that really gives us some insight into how we can live a long and healthy life? And the answer is undoubtedly no. This is her as well. She smoked up until the age of 119, it turns out. She drank it, she didn't exercise, she didn't do anything we think, at least we believe, it's sort of like that Woody Allen movie, we think are, of benefit. And so in reality, studying exceptionally long-lived humans has not turned out to be terribly insightful in terms of understanding how and why we age. And so the real, I think, the real change in the field, which has been evolving over the last 20 or 25 years, has not really come from studying humans. It's been studying from much simpler organisms. And the organism that really has really revolutionized our understanding of aging biology, in my opinion, and it's not something that I personally have worked on, but is the simple organism, like in this case, C. elegans, and various laboratories, Cynthia Kenyon's, Tom Johnson, Gary Rufkin's lab, identified in worms single mutations in single genes that could then extend the lifespan of worms in a reproducible fashion. And the exciting thing about these discoveries was that these genetic single gene mutations or manipulations are in pathways and genes that are completely conserved in all of us, suggesting that the lessons learned from these very simple organisms may in fact be applicable to how and why we age. So I've talked a lot about genes and everything. So I just want to ask the sort of simple question is, how many people here believe that their lifespan is predominantly genetically determined, that is based upon the genes that you inherited from your parents? Who believes that? Who believes it's mostly environmental? Okay, pretty even, maybe a bit more. Okay, so make it a little harder. So these are various traits so for instance, your height is about 80% genetic. Your body mass is about 50% genetic. So if you're tall, you can thank your parents. If you're overweight, that's your problem. So on this scale, from 1 being completely genetic to 0 being completely environmental, where would you put lifespan? Anybody want to give a decimal? 0.6, 0.7? Do I hear? Whatever. I feel like an auctioneer. Okay, but it's much lower than that. It's about, people, these numbers keep on going downward, but basically it's probably less than 25% genetic. I mean, in exceptionally long-lived people, that may not be the case, but based on twin studies, it's relatively little genetic component, not 0, but relatively little. So what are the other things that really are important? And one of those things, and the reason I don't eat lunch, is that if you look in organisms, very simple organisms, from yeast, certainly in rodents, that if you restrict the amount of food that they eat to the point of not starvation, but to the point of just the bare essential a number of calories that they need to sort of meet thermodynamic equilibrium. So in mice, that's about a 40% reduction. If you give the mice everything they want to this point, is about a 40% reduction in calorie intake. And across that entire reduction in food intake, the animals tend to have an extension of lifespan. And that's true for, as I said, very simple organisms like yeast, up to mammalian species. And if you look in primates, humans, it's a little difficult because it's hard to randomize long-term studies. But there are 2 large studies that took place, one at the NIH and one in Wisconsin, that came to slightly different conclusions, but there at least appear to be a signal that reducing energy intake or calorie intake in primates also extended lifespan. So now we're going to transition a little bit to the stuff I do. So again, check your phones if you're not interested. But so the question is, how does this work? Why is eating less beneficial? And my lab for a while was interested in 2 pathways that might regulate this process. And I'm just going to show you data from this mTOR pathway, just because I just wanted to hammer home this point about the regulation of aging. It's not that this is a great study or anything, but it is what it is. The mTOR is something that a lot of people here, I think, are interested in as well, but it's an intracellular kinase. It phosphorylates various proteins in the cell. And it itself its activity is regulated by the intracellular nutrient availability. So when there are lots of nutrients available, mTOR is active, and it does a lot of things in the cell to allow the cell to grow. And when the nutrients are scarce, mTOR activity is inhibited, and it again does a lot of things to protect the cell in the absence of nutrient availability. So it's really like a rheostat for the cell to sense intracellular nutrients and to make the cell grow or not grow based upon that. And the reason people got interested in for aging, because there's this program the NIH has where they test various molecules to see if they can make mice live longer. And it's about 4 or 5 molecules every year get nominated. And for a while, they really weren't having much success until they came upon this drug rapamycin, which is an mTOR inhibitor. And they were able to feed it to mice and show that giving this mTOR inhibitor rapamycin made this mammalian species mice live longer. And so it became this idea that you could potentially manipulate lifespan in a complex organism like a mouse with a drug, basically. So that was very exciting. And so we did this experiment in my lab when I was at the NIH, where we try to genetically reproduce this phenomena and then allow us to try to potentially understand how this was working. And so I won't go through the details, but we generated these mice that have about a 75% reduction in mTOR expression. And you can see those mice here. This is the normal mice, and this is the mouse that has reduced mTOR. And mTOR actually regulates cell size. So these mice were a little bit smaller than their normal compatriots. This actually, I like telling this story. So when we published this paper, I don't know why, but it got a little publicity, and actually it was written up in the Wall Street Journal, and they came to the lab. They usually do cartoons, but they wanted to take a picture of the mice. So it's actually very hard to get mice to pose for photographs, you know. So what you don't see is that my two postdocs, they're here holding the tails. But very embarrassingly, this mice peed here. I don't know if you can see it. There's a little green thing there, because they weren't used to being held in that unusual position. Anyway, so these mice are smaller, and they live longer. So they live, just like giving rapamycin, they live actually a little bit more than the rapamycin mice. They lived about 20% longer. But we did something where we analyzed these mice in a variety. So overall, the lifespan is increased, right? But the mouse model allowed us to then delve into each of the tissues and organs as best we could and ask, well, is aging a uniform process? So now we've done a genetic intervention where the mouse lives longer. But then if we look in each tissue, is there evidence that we've slowed aging in that tissue? And if so, is that true across all tissues or not? And the answer was not. So we did a lot of phenotype of these animals that live longer. And at the same chronological age, in most tissues, the animals that were going to live longer because they had reduced mTOR looked younger. They had better, more preserved youthful function. And here's an example of this in terms of a test for memory and learning. And so it's not important what the test is. Screw this up. But here, you know, the height of this bar is really the inverse of how quickly they can learn a task. And so when they're young, they're both the control, which is in black, or the mTOR reduced mouse that lives longer are very similar. But as they got older, the mouse that has reduced mTOR at the same age was able to learn and remember things better than the control mouse. And that all consistent with their brains being younger in a way, even though they were the same age. But there are various tissues like the eye or the bones that in the mouse that live longer actually age quicker than the control mice. And so that suggested to us two things. One is that aging is not uniform. And I think, you know, for people who take care of patients, that's actually a phenomenon that you know pretty well. You know people whose, for instance, brains are aging very quickly, whose vessels are not, and vice versa, right? And so, or bones are aging at a different rate than the rest of their body. And so this is a genetic model of an inbred mouse where we manipulated one gene that made the animal live longer, but each tissue responded to that in a different way. But it also, I think, raises this point about the regulation of aging, because again, there are 30,000 genes in the mice, right? We've manipulated one gene, and we're not even getting rid of that gene. We're just reducing its expression by about 75%, right? So it's about 25% of what normally is expressed. And changing that one gene produced a 20% increase in overall lifespan in that animal. Okay, so that if that was you, if your average lifespan was 75 years, if we could do this in you and it had the same effect, that would change your average lifespan to the age of 90. Okay, and that's just by changing one gene. Okay, so to me that says again that this is a regulatable process. It's not completely random, and that if anything that's regulated in biology or medicine is druggable and intervenable. And so I think that's an important, more than the study itself, important to remember. So how does this work? What is mTOR doing that that is important for overall lifespan? And the problem is mTOR does a lot of things in the cell, as I said, but I'm just going to concentrate on this one process here called autophagy, which mTOR regulates. And that's an interesting process that happens in all of our cells and tissues. And it's a process first described in yeast, and it won the Nobel Prize for the people who described it. But it's a process that is conserved throughout all evolution. And so it's something that's occurring in you as you sit there and listen to this as well. And it is an intracellular, in a way, recycling system that occurs in all of our cells and tissues that recognizes damaged proteins and damaged organelles in your body and wraps them in this membrane called an autophagosome. It recognizes trash in your cell, wraps it up for you, and then delivers it to a garbage disposal in the cell known as the lysosome, right? So it is basically sees trash, recognizes it, wraps it up like you would put your trash in a trash can, and then it brings it to the lysosome where it gets degraded. And then those materials that from the degradation of those damaged goods come out of the lysosome and are used to synthesize new undamaged proteins and organelles. And why is this important for aging? Because a number of, again, in simple organisms like the fly, have identified a number of interventions that are very disparate that increase overall lifespan in flies and worms. And trying to understand what could be common, I think the best explanation for what could be common is that all of these interventions appear to produce an increase in autophagy, an increase in this recycling system as a means to increase overall lifespan. And, in a way, if you look at old tissues and you see an accumulation of debris and garbage, the assumption has always been is that you make essentially more garbage as you get older. But what I think this genetics is telling us is that it's not necessarily that you're making more, but your ability to remove it and degrade it is impaired. And so it accumulates not because of more synthesis, but because of inefficient removal. Okay, so that's true in flies and worms. Is it true in higher organisms? And so again, a while back, we made this reporter mouse that allowed us to, for the first time, to visualize, in this case, a subset of autophagy known as mitophagy in animals. And I won't go through the details because it's not important, but what you can see here is the brain of a young animal, and this orange or red fluorescence is an indication of how much autophagy is happening in the brain. And this is the hippocampus where you're learning new things like those mice in that other experiment I just showed. And so you can see that in the young animal, there's quite a lot of this orange and red staining or fluorescence indicating high levels of autophagy in the brain. And in an old animal, that's all essentially gone. And so that and other things suggested to us that as we get older, this recycling system gets inhibited or it becomes defective. and maybe fuels this increase in intracellular debris and then therefore the decline in physiological function. So what is fueling this impairment? Why do we have this decline in autophagy as we get older? And again, we became interested in this over the last few years, and we concentrated on that last step, which is the lysosome, which is this organelle, as you know, that's involved in degrading a variety of material that comes into the cell and reducing it back to sort of biosynthetic intermediates. And there was a lot of evidence that suggested that the ability of the lysosome to function as we age gets impaired. And so we became interested in trying to understand how does the lysosome repair itself? And, what is the process for that happening? And I won't, well, okay, I'll do a little bit of this because it's really, anyway, so we said, okay, we don't have any idea how the lysosome repairs itself, although there was a few things out there that suggested it. But so what very talented person in my lab, Jay Tanner, now has his own lab at our institute, devised was a screen to look for what happens to the lysosome when it's damaged. And I won't go into too much detail, but what he did is to generate this cell line that had this enzyme on the outside of the lysosome called a proximity biotinylation enzyme, a fancy term. But basically what it does is it is a enzyme actually from bacteria that takes biotin, which is like vitamin B12, beta, one of the vitamin B, I don't remember which one, but it's a molecule that this enzyme can take and covalently add it to any protein that's within 10 or 15 nanometers of this proximity biotinylation. And so that biotin tag is a useful tool in the lab because there's another protein called streptavidin that binds to biotin like an antibody almost in a very high affinity reaction. And so you've now coated the lysosome with proteins on their surface with this biotin tag, and allows you to then rapidly purify them from the cell using this other protein called streptavidin. So what he did is identified all the proteins on the surface of the lysosome under normal conditions, and then he damaged the lysosome and waited 30 minutes, and then asked, what are the new proteins on the surface of the lysosome that came there, okay? And the answer is there's a lot of new proteins that come here, and I won't go through all of them because we don't have all day, but he identified these several proteins and showed in green that suggested interesting pathway that might be responsible for repairing lysosomes. I'm just going to walk you through this just as an example of the kinds of things that my lab is interested in and I think are important for aging biology. So the first step in this pathway is the recruitment of this lipid kinase called PI4K2 alpha. And this lipid kinase phosphorylates a phosphoinositide in the membrane of the lysosome called PI and makes this new lipid called PI4P, okay? But normally there is none of this PI4P on the surface of the lysosome. This gets to this sort of theoretical issue, I think, in cell biology, that if you have an organelle like a lysosome or a mitochondria, you have hundreds of these in your cells, right? And if one of them gets damaged, you've got to identify the bad lysosome or the bad mitochondria from all the good mitochondria if you want to repair it, right? Or the good lysosome. And so you've got to mark that organelle with something that says there's been damage here. And for the mitochondria, it's a different signal, but we think for the lysosome, it's this generation of this new lipid on the surface that's normally not here, that serves as a signal to the cell that this lysosome is damaged and the other lysosomes aren't. And the reason that serves as a signal, because it recruits these proteins called ORP proteins that bind to that PI4P and actually bring two organelles in the cells close together. In this case, the damaged lysosome with the endoplasmic reticulum. And these are called tethering molecules. And so That's the second step that happens in this pathway. And they not only tether the lysosome to the ER, but then they exchange this lipid PI4P for another lipid called phosphatidylserine, which then builds up on the membrane of the lysosome and recruits this other protein called AGG2, which acts to transfer lipid between the ER and the lysosome, which is the third step. I went through all of that so I could get to the acronym that we use to describe this pathway, which is the PIT pathway. Our university, in a shameless attempt to get money from our dean, which didn't work, unfortunately. We got nothing, bupkis, as my grandmother would say. But anyway, that stands for the phosphoinositide initiated membrane tethering and lipid transport pathway, which we think is involved in lysosomal repair, and we think is involved in the age-dependent decline in lysosomal function, and we're trying to pursue that using various models. I bring this all up to then just to really go at what I think is a sort of a paradigm shift in biology, which I think you probably have all thought about or experienced in some way or the other, in that when I started my lab, I always was trained to sort of come up with a hypothesis about how things work, and then to go into the lab and design experiments to test that. So the aim, hypothesis first, fire, get the data. And I think biology is changing. I don't know if it's good or bad, but we had no idea, for instance, how lysosomes repair themselves, right? I mean, we didn't have a hypothesis, right? But what we did is we started with an unbiased screen, this proximity biotinylation screen. And as you can see, we got those mass spec curves, we got hundreds of proteins. And from that list, we came up with a hypothesis about how lysosomes repair themselves. And so, I would argue this is the opposite approach, and this is how a lot of science is going these days, is that you fire first, you do omics, you do a screen, you get the data, and then you aim, basically. You then come up with your hypothesis. And again, I don't know if this is good or bad, but it definitely is a different way of doing science than when I was a resident and thinking about a career in science as a physician scientist, how we would go about working in a lab. And so I think a lot of science now is driven by technology and by the ability to collect large sets of data and then to infer hypothesis from that data. And again, I don't know if that's progress or not, but that's the way things are. How are we doing for time? Yeah? Okay. So that's a little bit about the history of aging biology over the last 30 years, a little bit about what I do. How can we make this really impactful for people? So again, as mentioned, we started this Institute of Aging Institute, and we've grown to about 13 principal investigators. And the whole idea was that these principal investigators would study the basic biology of aging, try to understand the mechanism, try to identify targets. We set up the ability to do sort of high throughput screening against some targets and try to develop small molecules that might impact the aging process. And we wanted to develop a clinical infrastructure to be able to test either repurposed molecules or molecules hopefully we develop ourselves to intervene in not necessarily extending lifespan, but prolonging health span in individuals. And obviously that's a long process, and we're only 7 years into it. But I just want to give some examples of flair for the kinds of things that we're doing. So one of the things that my colleagues and I are interested in is this NAD biology. So some of you may know that as you get older, levels of NAD appear to decline in your cells and tissues. And there's some evidence to suggest that if you increase NAD levels, that you can restore more youthful biology, at least in various animal models. And in fact, if you go to Amazon, you can now buy various products to boost your NAD levels. And these are not NAD itself. NAD is charged. It doesn't get into any of your cells and tissue. So these are various biosynthetic precursors to making NAD in your body. And what people are selling are a variety of a different biosynthetic precursors. There's only one common thing among all the products being sold, that they're all like $49.99 a month, basically. Otherwise, biochemically, they're all very different. Prior to that, people said if you drink red wine, you get a lot of this molecule called resveratrol. And resveratrol doesn't boost your NAD levels, but it sort of, in a way, makes the NAD in your body and cells and tissue perhaps work a little bit better. None of these things were terribly satisfying, as a, true pharmaceutical. These are all sort of nutraceutical approaches that, are not, for instance, many of the precursors that you take for NAD boosting have a half-life in your plasma of about a minute or two before they're degraded. So the idea that they're going to really get into your body and change your NAD levels, it's not impossible, but it just seems unlikely based upon how most drugs work. And so again, I won't go into it, but we've developed a strategy that, we think is a much more, targetable way to increase NAD levels. And so that is by manipulating this enzyme in your cell called NAMPT, which is the rate-limiting step in NAD salvage synthesis. And so with this small molecule at sort of nanomolar concentrations, we're able to increase NAD levels in cells and tissues in mice in a very reproducible fashion. And this is a molecule that has, 8-hour half-life as opposed to 2-minute half-life. And the magnitude of increase, although it's, only doubling NAD, is really actually, we think, sufficient to combat the age-dependent decline, which is about 40% from young to old. So doubling NAD is more than enough in terms of reducing sort of more youthful levels. And so, again, we've been fortunate to, my colleague Bill Chen and Stacy Rizzo got a large NIH grant to actually get this into the clinic for cognitive decline, which is another avenue that NAD decline has been linked to. This is an area I just wanted to make you aware that we're actually not doing, but I think is actually pretty interesting, which is this idea of modulating the epigenetic makeup of cells and tissues. So as cells age, the DNA itself doesn't change that much, but the chromatin, the protein coding around DNA, called the epigenome, changes. And so there's a number of groups who believe that these epigenetic changes are what's really driving the aging process. And as some of you know, about 15 years ago, there was a Nobel Prize awarded for what are known as the Yamanaka factors, which are these set of 4 genes that can epigenetically reprogram any cell back into a stem cell. And they do it by epigenetically reversing let's say, the fibroblast back into a stem-like cell. So there is a way of erasing the epigenetic memory of a cell using these Yamanaka factors. And so there's a number of people very interested in playing around with these factors as a way of, again, basically resetting the biological epigenetic clock back to a more youthful state. Again, there's some evidence in cells and mice that this can work and can make younger tissues from an older animal, but it's not without risk. And so the question is, can this work? And I think the answer is yes. And I'm going to give you, I think, the best example of why it can work in humans, which is this process, which you may recognize as fertilization. So again, you start with, when you have a fertilized zygote, you have a, depending upon the age of the father, a 25 or 30-year-old sperm, and depending upon the age of the mother, a 25 or 30-year oocyte. They come together, and the magic happens, right? We won't go through the details that, you ask your parents, but they come together, and what comes out of that after 8 or 12 or 16 cell divisions is a embryo that's age 0, right? So the components were in their 20s and 30s. The result is an epigenetic reprogrammed embryo that's 0, right? So that says that it can work in this example in a safe way. The problem is, can we do this in an adult somatic tissue? And the answer is we don't know. But there's a lot of billionaires who've invested in large companies now that are trying to do this. It's been a while now since they put their billions in, and they're still the same age, so I don't know. But there are people working very hard on this thing, and I just thought you should know about it and think about it because, as I said, there is precedent that this could work. Actually, the reason I'm down in Florida, one of the reasons, not to just be with here, but my daughter just had a little baby girl, so I just became a grandfather. So this process does work, and she's beautiful. I just have several hundred pictures I could show you in case you're interested. The other thing that, if we want to do interventions, we can't obviously do things in humans and wait 30 or 40 years. So we need ways of testing interventions, and we need surrogates for whether or not those interventions are going to work. And I just want to highlight one recent study that a colleague of mine at Pitt did, Aditi Gurkar, and she was on Science Friday, so you can listen to more of it if you're interested in. But she took two whole cohorts of patients and tried to find a metabolomic signature that would distinguish them. And the two cohorts were what you would call healthy agers. These were people over the age of 75 who were very robust, versus what you would classify as rapid agers, which were people under the age of 75, between 65 and 75, who were sort of pre-frail. And then try to identify in this cohort, and then in a subsequently replicative cohort, whether or not there was a metabolomic signature that to distinguish unhealthy from rapid from slower ages. And again, you get a lot of things when you do this thing, and she got this signature of metabolites that appeared to signify poor rapid aging compared to healthy aging. And it's these types of signatures, these types of fingerprints that we could potentially use as ways to see whether or not any given intervention is affecting the rate of aging, rather than rating the 20 or 30 years that we would have to under normal circumstances. So these and other approaches, I think, are going to become really pressing the more that we have the ideas, the targets to begin to intervene. And so just in terms of intervention, I just want to talk about a trial that we have initiated at Pitt that now has enrolled about 25 elderly subjects on the way to about 60. And again, just to give you an example of how we can potentially leverage, sort of the basic biology of aging to therapies that might impact the rate of which people age. And so this was a trial, again, targeting, in this case, a phenomenon known as inflammaging, which is this activation of the innate immune system as you get older. So as we get older, a variety of epidemiological evidence suggests that there are increase in sort of sterile inflammation, the activation, again, of various cytokines that appear to be induced in the absence of any sort of known infection or disease just by aging itself. And the cause of this inflammaging is really unclear. It could be the accumulation of senescent cells in our body that secrete a lot of inflammatory factors. It could be the leakiness of our gut that lets bacteria into our bloodstream as we age, and that could be triggering this immune response. It's unclear. But a number of inflammatory cytokines have been linked to this inflammaging, and inflammaging has in turn been linked to worse prognosis, increased mortality, risk of hospitalization, risk of nursing home admissions. And of all the factors, looking over that literature, we thought that an increase of interleukin-6 or IL-6 was the strongest factor linked to frailty and mortality in the elderly. And there are, again, listed here are a number of studies that have linked increases in IL-6 with frailty, and there's other studies as shown here, going back to my interest in cardiovascular disease, a huge literature suggesting that a rise in IL-6 is linked to age-related cardiovascular events as well, but same thing with cognition, et cetera. And so what we've done with the help of actually a variety of philanthropic supporters, as well as the support of CSL Behring, which has developed this IL-6 antibody, we're randomizing patients over the age of 70 who have high IL-6 levels for no apparent reason except for age, to placebo versus an IL-6 antibody over the course of a year, and asking whether or not reducing IL-6 in the patients that get the therapy makes them function better, think better, their vasculature improve, et cetera. And so these are sort of some of the sort of criteria and dosing strategies. And the primary endpoint is really just gait speed. But again, as I mentioned, we're doing a number of sort of exploratory studies to ask, where does reducing inflammation in these patients benefit them the most in anticipation for larger studies, which might be empowered to be able to define these in a more meaningful way. So let me just summarize what I wanted to get across. Oh, good. So takeaways. Age is the biggest risk factor for most diseases. There's some good things about getting older, like having grandchildren, but otherwise, there's a lot of landmines, unfortunately. But the good news is that it's regulated. I hope I've convinced you that. And again, I think this idea that it's, again, when I was, at your stage in medical school or even before, I didn't think of the regular nature of aging. And I think, again, anything that we can understand, anything we can understand the mechanisms that drive it, that suggests that there's a way to target it. And again, I do think that there will be strategies to slow the rate of aging and therefore slow the rate and progression of a variety of diseases. And that's what a lot of people are calling this neuroscience hypothesis. I think, does that mean we can all smoke and drink and still live to 122? I don't know, but who knows? Maybe at least 122 without the smoking and drinking. And let me just end by the imperative nature of this challenge, because we are getting older as a society, and so this is demographics that I think are a little bit frightening in a way. So throughout all human history, the number of people under the age of 5, as shown here in the blue bar, compared to the number of people over the age of 65, has always been in the sense that there have been much more young people than older people. So here's data from just 1950, right? That 5% of the world population was over the age of 65, and 13% was under the age of 5. And about, I don't know if you noticed it, but maybe 4 or 5 years ago, these curves crossed for the first time on the planet. And now, as you can see, the curves are going in the opposite direction all throughout the world. And so that by the year 2050, just 100 years after this curve was shown here on the right, these curves will have completely inverted, and the number of people over the age of 65 on the planet will vastly exceed the number of people under the age of 5. And so I would argue that it's probably a good time to understand why and how we get older. Not to say we want more old people, but we want people's lives to be as healthy and robust as possible. And I think understanding these mechanisms will help. Let me just thank some of my colleagues whose work I mentioned. Jay, whose work on that PIT pathway, has his own lab now. Stacy and Bill, working on this NAD additive, who I talked about, and Ann Newman, who's running our clinical trial on IL-6 reduction, and our various funders. Thank you very much.
Thank you, Dr. Finkel. That was an absolutely superb grand round. So I'm sure we have lots of questions. Maybe I can begin in your paradigm of decreasing energy intake and that relationship to aging. What about stress? How is that related, at least in the laboratory? How can you look at that, and what arm of the pathway is that involved?
Yeah, so, you know, I mean, the stress means different things to different people, and resiliency means different things to different people. So, you know, one of the things that we've been very interested in over the years has been this idea of hormesis, which is that if you have a little stress, that actually prepares you, and you take a variety of steps in the body that allows you to withstand larger stress that come down the road. So stress by itself, I don't think is necessarily a bad thing for aging. I think exercise, um, starvation are all stresses that in turn provoke responses in the organism acutely that persist beyond the stress that actually are a protective mechanism overall for aging. And so even for mutations that in the worm, for instance, that reduce, increase lifespan, they all, you know, the original mutations were called DAF2, DAF16. They're all, the D in those mutations stands for Dower, which is a phase of the worm where when it faces an environmental stress, goes into this sort of alternate phase known as Dower. That's sort of like what we would think of hibernation in larger organisms. And the mutations that increase lifespan didn't actually go all the way into Dower, but had a lot of those sort of protective sort of mechanisms that you see in the quote Dower phase. And activation of those protective mechanisms, those stress pathways, actually conferred lifespan extension. So the long and short of it is that I think things like exercise, which are actually very beneficial, produce a lot of damage in your muscle, produce a lot of stress, produce oxidative stress, all the things that we've linked to aging, but they do it in a discrete fashion that allows the cells and tissue to see that stress and activate a variety of countermeasures that actually persist beyond the stress and actually may be beneficial.
Dr. Bernal.
Hello, this is Dr. Ashfaq. Can I ask a question?
Actually, we have people in the room first that
Okay.
raise their hands. Thank you.
Sounded like God, I didn't know. Yeah. Yeah, or yeah, tissues, but yeah, yes. Yeah, yeah, yeah.
So when you find all these things, I wonder
Yeah.
how you
Right. Yeah, so, right, I think that, you know, suggests that there will probably be no intervention that slows aging everywhere. Oh, so the question is, if in the mTOR model, we saw that the animals lived longer, but that certain tissues were better and some tissues were worse. And so how can you direct your therapies to the appropriate tissue? And I think my answer is that you probably can't, and you probably will not be able to find a single intervention. For instance, if you had a really good mTOR inhibitor, I would argue, based upon our mouse work, and again, you know, you can't necessarily extrapolate from mouse to human, that if you really had a safe rapamycin-like analog, that, you know, based on our mouse model, the people who took that would think better when they got to their 80s or 90s, but they would suffer bone loss and maybe, you know, and cataracts more than their thing. So you'd have to, you know, think of some sort of cocktail for your bone that would be different from your brain, basically. And so it will get undoubtedly complex.
Okay, Dr. Ashtak online. Yes.
Hi, thank you so much. I'm a nephrologist and I can suggest a good model to study aging. Patients with chronic kidney disease, actually, most of them die of premature aging, as you know. And there have been a lot of things which are associated with this. One is the clotho deficiency in patients with chronic kidney disease approaching end-stage kidney disease. And the second thing is that most of the chronic kidney disease patients are in chronic stage of inflammation, and not just IL-6, but other inflammatory cytokines are upregulated. So these are, do you have any thoughts on these things?
Yeah, I mean, I think you know, in terms of patients to intervene, in terms of improving health span, I mean, I think, you know, our study is in completely healthy people. You know, it turns out to be very difficult to find people over 70 with high IL-6 who have no issues, basically, which is the entry criteria. So we've screened thousands of people to get to 25 people. So, I mean, I think we have to rethink, you know, whether or not a model of healthy aging versus disease, a patient population with a disease state that predisposes to rapid aging would be the best, you know, place to start these types of interventions. But so, I mean, I think chronic kidney disease is certainly on the list of conditions that, you know, you might think about where you can think about aging interventions as a way of treating the underlying condition. But yeah, so obviously, you know, when the patient has other medical issues and the intervention, you know, the safety profile of the intervention becomes, you know, what you ascribe to the medicine versus what you ascribe to the disease would have to be thought about.
Thank you,
Thank
Dr.
you.
Finkel, and congratulations on the welcoming into grandparenthood. It's definitely an important stage and a pleasurable stage in one's life. I want to remind everybody that the Department of Medicine Research Day continues with posters outside on the scientific quad until 4:30, when we will announce the winners of the Shark Tank, as well as the abstracts and other presentations. I want to remind everybody to check the QR code on your exit or on the web to get CME and MOC credit. Again, thank you for a wonderful grand rounds, and everybody look forward to seeing you the rest of the day.